Okay, I think we're about ready to get started again. If everyone could take a seat, we'll get going. So, kind of wanted to remind everybody, particularly I know there's a few of you that weren't here yesterday, the way that we're generally running these discussions is we'll start off with a set of comments and a question and answer session between the panel members and the consultant, and then following that we'll have an opportunity for the public to come up and make comments and ask questions. As a reminder, if you're coming up, this is going on TV and going over the internet, so we need to have you come up to the microphone, state your name and affiliation before you ask your question. And I'd like to encourage people that are watching this these panels over the web to submit questions if they have something they'd like to ask either our panelists or the consultants. So let me start this interaction between our panel members and John Houghton by briefly reintroducing our panel members for those of you who weren't here yesterday. I gave a fairly terse introduction yesterday, and I'll be even terser today. So starting off on the end there, Cy Simonstad is a professor at the School of Aquatic and Fisheries Science at the University of Washington. He's the coordinator of the Wetland Ecosystem team. Next to him is Dr. Jeep Rice at NOAA Alaska Fisheries Science Center in Juneau. He's the program manager for the Habitat Assessment and Marine Chemistry group. Next is Mike Stone, recently retired from his position as Fisheries Division Chief for Wyoming Game and Fish. And last, Hal Geiger, currently with the St. Hubert Research Group in Juneau, but retired not too many years ago with a long career as senior biometrician with the Alaska Department of Fish and Game. And so, Cy, you're one of the people that's been assigned sort of lead reviewer responsibilities in this area, so I'd like to throw it to you to make comments and ask John any questions you'd like to.
Sure, thanks. First of all, I really need to thank John for a phenomenal digestion, or I don't know, maybe regurgitation of a huge component of the studies. If you haven't read the documents, it's like drinking from a fire hose. But thanks again for a good synopsis. And I also want to sort of predicate my perspective on a little bit of, I think, a difference from what we've heard about from the mine area studies, the Bristol Bay drainage with the Cook Inlet drainage, in that I think the objectives are a little bit more focused. The stated objectives are to characterize the fauna, flora, and habitat conditions in marine areas that could be affected by the development of a port and related activities. And the options of where the port and the transportation corridors go is a little bit more focused and a little more limited. It still covers a large area, as John indicated, particularly for the fort, but the transportation corridors is perhaps a little bit more focused. And so my idea is that the objective should really focus on what we already know and what can we do to fill the gaps. So I sort of hoping the document and John can give us some ideas about the gaps that really need to be filled. And those should have been translated into questions that really anticipate the EIS NEPA baseline needs. And that should be reflected again then in the study design. So things I'm going to sort of focus on are things where I have some questions or concerns about the study designs. I mean, the interpretations, I don't think anybody expected major interpretations. But again, the gaps are perhaps the most valuable component of those interpretations. So I've got a few things, and maybe John can respond to each of three or four. One is the habitat mapping, and I guess given my background, I jumped immediately to look for the eelgrass maps, because that was a stated objective that John described, and actually described some area coverage. But I really couldn't find either the methodology or the maps that identified the spatial distribution, the concentration, or you mentioned patch sizes. And given the fact that eelgrass is important herring spawning substrate, at least in certain parts of those two estuaries, is likely a major organic matter source for at least the benthic in-fauna invertebrates, if not others in the system. Really good question is that data available, and did I miss it?
Okay, well, we have done some work after 2008. We have produced some reports of that work, and in fact have approached eelgrass mapping in a couple of different ways. One was by aerial photography and with ground truthing and interpretation of the photography, and the other has been by foot surveys, basically walking, delineating areas with substantial eelgrass. So we have developed some preliminary maps of that stuff. I mean, it clearly is a data gap in the 2004 to 2008 report, and I think we've done what's needed to fill it. Unfortunately, it's not in the presentation at this point.
Great. I just hope that is brought forward. I guess another one is the marine benthos. I've known John for a long time, and I know his heart's in the rocky intertidal and gravel cobble systems. But I'm sort of concerned about the study design sort of lack of focus on the soft substrate parts of the system, particularly the inner bays like Iliamna Bay and the Williamsport area, which may have a concentration of the transportation corridor, the pipeline corridor. And I think you'll agree is soft sediment communities are much more vulnerable to a variety of stressors from that. And as far as I could figure out, there really were no benthic in-fauna samples in those big, large mud flats in the inner bays. And so, again, did I miss something, or is that sort of disproportionately not as well covered as the rocky intertidal? Also, sort of was trying to figure out were the benthic infauna and epifauna of eelgrass specifically looked at. As you well know, there's a number of taxa of macroinvertebrates that are specifically associated with eelgrass that may in fact be important prey of fish species, which I'll come to later. So I'm trying to figure out is in addition to your substrate variability, which you can link to variability in the macroinvertebrate community, is there specific data on eelgrass fauna that would be able to identify as you suggest, the links between not only the fish and the invertebrate community, but also the habitats that are unique to those invertebrates? And it looked to me, as far as I could figure out, is there wasn't a necessarily consistent tidal elevation array. That some sites were mid elevation, some sites were lower elevation. There wasn't sort of a classic intertidal gradient to that, which again it would be good to have on a site by site basis. And of course, the other thing you'll probably be used to me hearing is what happened to the myofauna, which again are quite often the component of the prey community. You're using 1 millimeter sieves, most macrofauna is half, and there's it's not surprising you didn't get very many of the prey organisms in there. And is that something that should be considered to make that link, that food web link, better?
Okay, there were quite a few pieces there. And all good questions. The first part you mentioned was in the benthos design. Was there sufficient emphasis on soft substrate sampling, and particularly in the inner bays and transportation corridor? And that's something that we have recognized, having begun to understand more fully what those facilities could look like, and we have added some stations, particularly in Iliamna Bay. We haven't done a lot of work in the innermost part of Inniskin Bay because no scenarios we've seen have or would result in changes in that area. But we have added a number of stations within Iliamna Bay, and have some data, in fact, that are being worked up. And I'm not sure what the schedule is for making those available, but they will be there for the EIS process. Let's see, you asked about the specific infauna and epibiota associated with eelgrass, and we haven't done anything targeted specifically at that. If indeed we have a scenario where eelgrass would be directly affected by the project, then that would certainly be something to consider. So we shouldn't lose sight of that. And one thing I might note is that these eelgrass beds, because of the ice influence that I mentioned, they look quite different from eelgrass beds in Prince William Sound or Puget Sound, where the beds and the root structures are much more stable and long-lived features of the environment that allows them to develop a more targeted association of species. If you look at these beds, you see eelgrass. There's very little epibiota on the eelgrass blades itself, like we have in other older beds. The plants are uniformly small, seldom exceeding maybe 30 centimeters in length, and relatively narrow. Some of them are diminutive enough that we took a close look to make sure it wasn't Zostra japonica. But anyway, but a good point, and something that probably should be included in future work, where once a development scenario becomes clearer. Then finally, I think you asked about myofauna, critters smaller than 1 millimeter, and I guess we selected the 1 millimeter in 2004, when in fact Steve Jewett and UAF was running that piece of the program, and have perhaps blindly stuck with it. But again, that does add a dimension that we don't have in our current data set.
So not too much on the nearshore fish, because I think you've really done a fairly impressive job in terms of effort and particularly temporal coverage and repeated sampling. I guess the one thing I do wonder about is whether you followed up, given the fact that you've got two other major forage fish, surf smelt and sand lance, that very likely spawn in that area, is that there's established protocols for identifying their spawning sites on the beaches. Are you picking up that protocol and applying it to those appropriate beaches?
Yes.
Great.
No data yet, but we have done that, and we recognize that surf smoke, in particular, based on our catches in the beach seines, it sure looks like they should be spawning sometime in the probably early summer in that area. And we did collect a great many gravel sand samples and have looked for them all and can't find any eggs. But the habitat, when you get right down to it and look at it, isn't like the classic Puget Sound forage fish spawning habitat. It tends to be much coarser, and it's very limited areas where we can find finer materials that you would think they might spawn in. But we did put out a pretty good effort to try to do that without success so far.
Great. So then the last one is this thing called food web relationships. And I'd really like to hear why and what the rationale for that is, because at least as represented, let's see, how can I be polite on this, is really fairly insufficient for a number of standpoints. I mean, first of all, are the sample sizes. Shouldn't really characterize food relationships by sample sizes of 8 and 6 and 10, and even 47 chum salmon. If you're not sort of designing and stratifying it over, space and time, is extremely hard to interpret. And I guess I don't understand when you've captured, what was it, almost 4,000 chum and 3,500 pinks and 75,000 herring, that you end up with sample sizes of 47 and 10 and 12 and 6. And I guess what I hope is that it wasn't reflected that way, and the document is just a preliminary view of what you really have to draw from, and those samples are emerging with sort of stratification to give you an idea of temporal, spatial, and habitat differences. But the other thing is I also hope that the taxonomic resolution is going to be more informative. I mean, right now, given that resolution, I can't tell where those fish are feeding in the water column, or they're feeding on the intertidal, especially something like copepods, which could be everything from planktonic copepods to parasites. So again, I hope the resolution, again, the idea is to characterize how that nearshore system is potentially supporting key species through food web linkages, that resolution is going to emerge.
Well, what can I say? We have continued to do more of that kind of work, but probably not to the degree that you're suggesting. And I'm not sure there's a justification for it, but that is what we have done, and obviously more than it has been reported in my slide presentation, but not probably to the extent that you've described. So there again, that could be something to look at down the road.
Especially if you think that eelgrass is a prominent habitat player in that system, which has some very unique fauna sometimes.
Yeah.
I've got enough concentration of that. Open it to other panel members.
Yeah, so would you like to go next?
Sure. I guess first of all, I'd like to compliment you on 9 months of sampling, for example, which went from March through November. It's I'd really like to see a complete year, but that's covering 9 months of it. So I think that's a pretty good deal that you've done there in that respect. I guess I won't get asked too many questions in a sense. I just make a few comments. One, I would hammer the eelgrass habitat, the mapping, and what's the significance in the future. And I think it is a fragile and vulnerable habitat that is important. So mapping it and watching how it fluctuates from year to year to year, even if the port is some distance away, vessel traffic and increased activity and development and all those sort of things make that habitat vulnerable and fragile. So I'd like to see that aspect bolstered up. As far as lots of fish studies there, I kind of sometimes wonder why would I care? I do care about the forage fish, so and spawning, any spawning activity of those forage fish. There's not the habitat is limited, I suspect, for both spawning and rearing habitat of those forage fish species, and they impact the environment pretty significantly. So in a way, going from presence to and absence type surveys to more focused surveys on where those species spawning, where the early life stages rearing, how does that relate to the future port development? Those are concerns I have in terms of the future in that area. And with that, I guess I'll pass it on.
Okay, Mike, go ahead.
I don't have any.
Okay.
Well, perhaps I'm getting ahead of myself, and that might be just the answer to your question, but that might be your answer to this question. But as I sat there and I looked at the inventory, I know that your talk was intended to be kind of an inventory of what's there now, but I've just found my mind constantly drifting off to, well, what's really important here? So I guess I would like to ask, so now that you've completed this, what's really sensitive here, and what could be something we would worry about as far as a port facility? For example, a spiny dogfish, I would guess, would be something you'd be relatively unconcerned about, and there'd probably very little chance a port facility would have a big effect on those. And even if it did, there's probably lots of spiny dogfish in Cook Inlet, so you'd still not be worried. Mm-hmm. But what is it that you've learned? And if that question's too early, well, what could you tell us about the process that you're going to go through to develop more hypotheses and so forth?
Well, I think clearly forage fish spawning is one activity that's very spatially specific. And if there are facilities proposed in areas that are documented to have been used for forage fish spawning, then that certainly is an important aspect of impact assessment that demands having a certain level of data available to respond to. So that's clearly one thing. Cy brought it out, and Jeep did on the species other than herring, where we've not yet figured out if or where they spawn within the study area. An aspect of the herring spawning, to date we have only been looking intertidally for that. There's a potential that there's sufficient algae in some of those areas subtidally where there could be forage fish spawning that we haven't yet observed. So that's one aspect that probably needs more work once a specific facility layout is identified, certainly. For the eelgrass and spawning on that, there is a clear, in the surveys we've done, we've seen a clear lower bound to both eelgrass and the herring spawning activity on that eelgrass. Doesn't appear that either of those things extend subtidally, and that's probably a function of the water clarity in the area that doesn't support eelgrass below a certain elevation. But yeah, so anyway, that's one aspect that's certainly important. Let's see. I think overall the function of the tide flat could be important, depending on the extent of, again, facilities that are proposed that would directly occur on the tide flats, and that's in part why we've added sites that I mentioned in my response to Cy earlier. So we're getting a better handle on that. Let's see. The juvenile salmon rearing is obviously important along these shorelines. So the nature of changes that could impact that will perhaps indicate additional types of work that needs to be done. I think we have a pretty good handle on the timing of those out-migrating runs. And with the reservations expressed by Cy, we know what they're doing as far as feeding in those areas. They're in the deeper water areas. The things that have been struck me, I guess, as particularly interesting are and that didn't necessarily come out in my presentation. We do get a fair number of juvenile halibut in our trawls, ranging from very small to moderate ping pong paddle size halibut. Understanding where their habitat use occurs and how they're using the habitat, particularly if there's features that would impact some of the deeper areas. That's something that certainly could be considered an important resource just because of the regional significance of the halibut. Tanner crab is another one that I did mention, that they are not only linked to halibut but also obviously important by themselves. So those are a few things.
Any follow-up from panel members? Anything further?
I just one more comment, and that is that you recognize, of course, that because there's no commercial fishery on herring over there in Kamishak Bay, at least that's the projection, at least for some time. That means there won't be very many ADF&G resources spent toward trying to evaluate that population and the spawning and tracking it over time in years. So that's kind of bothersome, I guess. That hope you guys are able to pick that effort up a little bit.
Anybody else?